on behalf of Proteus. My name is Abhishek, and our faculty speaker today is Dr. Swagatika from Department of Chemical Engineering at IIT Madras. So she did her PhD in the field of systems biology from the University of Iceland in 2014 and carried out her postdoctoral work at the Luxembourg Centre for for Systems Biomedicine between 2013 and 2015. So Dr. Swagatika's group studies extensively in the field of constraint-based metabolic modeling, and she uses these techniques to study metabolic disorders, neurological disorders, and cancer. And Dr. Swagatika was awarded the Inspire Fellowship by the Department of Science and Technology, Government of India in 2015. And I was lucky enough to work with her in my first summer, that was back in 2018. And I had a really enjoyable research experience. And personally, I can say that I was really lucky to have Swagatika ma'am guide me in my first, let's say, venture into science, and it was really enjoyable. So I'm quite excited for this talk, and I'm sure it will be a great experience for all of us. And I would like to hand over the mic to her and request everyone to mute themselves and keep their videos off until the interactive session at the end. So please, ma'am.
Okay, thanks Abhishek. Thanks Proteus, the Biology Club at IISER Trivandrum for having me come today and give this talk. So I was pretty surprised when the invite came, but also excited that yes, the enthusiasm is not down during the COVID period. So yeah, so today I will be talking about metabolic modeling and how we can use that to study different diseases, as well as what are the established models that we already have, the pre-existing ones, which we can take use of. I'll also give a practical example as to how was it used to study one of the most prominent cancers within the Indian population. Okay, so what I'll do right now is I will switch off my video, but I'll turn it on again during the question answer session. Okay. Okay, fine. So that's what the title says. We'll be discussing about the metabolic modeling and, of course, how we can use it for the personalized medicine, how we can generate patient-specific metabolic models. Okay. So before that, let's talk about the different outlines or the contents of my talk today. I'll give a very brief introduction on what exactly is systems biology, what exactly is the bridging stone that systems biology is between computational versus the experimental world, how systems biology brought these two together, and we can use this to our fullest advantage to study different disease mechanisms. Okay. Then I will discuss about the most comprehensive human metabolic network, as well as a very short introduction to constraint-based modeling. What is it? What is the fundamentals behind it? I'll give a test case about how we can develop very specific patient-specific models pertaining to retinoblastoma. In this case, it's a cancer of the eye. Okay. After that, we'll venture into whole body metabolic modeling. That is how we can use the most comprehensive metabolic network of human to derive not only one model, but rather for the entire human body in an integrative manner. I'll also show you certain of my research themes, as in what are my current research areas that I'm involved in. And of course, you guys are free to join in any of the areas that you wish to pursue in future. Okay, so the evolvement of systems biology rather comes from the existence of various data generation techniques. Just like we have the central dogma, that is the flow of information from the, excuse me, I'll just take the pointer, it becomes easier for me. Okay, yeah. So we have the flow of information from the levels of nucleic acids to the level or to the language of proteins, and finally we have the downstream metabolic transformation, or in other words, put together metabolism or cellular metabolism. Okay, we have this nice linear pathway. Subsequently, we have the techniques that are used for measuring each of these molecules. Okay, we have genomics, or here means the open reading frames. Okay, for measuring the level of mRNAs that are expressed in a cell or tissue type, we have the transcriptomic data. Similarly, for the content of the proteins or the whole cell proteins that are expressed can be measured by the help of the a technique that is called proteomics. And the most recent form of omics data that is available, it is called the metabolomics, okay, where you measure the amount of metabolites across different biofluids. By biofluids, what I mean is it could be blood, it could be CSF, it could be urine, sweat, tears, okay. So these are the different omics data available. Since we have the availability of a humongous amount of omics data, rather the 15 years behind, we had the entire biological science dedicated towards data generation techniques and generating the different kinds of cell-specific or tissue-specific data, not only for human, but also for different kinds of living organisms, okay, typically for the microorganisms. So how do we explore this data? How can we know what is this data trying to tell us? That's where systems biology comes, okay? So this is the time to venture into systems biology, because there is a humongous amount of information flow in the form of data. We can use the tools of systems biology, typically the constraint-based modeling that I'll tell about in a few more slides ahead, how we can use that to explore this data types, okay? So that's what systems biology does. It not only explores the different kinds of data, rather it integrates everything onto a level of a model, okay, to capture their interactions. So you'll have to imagine that a cell, not only, so if I go back again to my previous slide, a cell doesn't comprise of these molecules individually. Rather, there is a nice crosstalk or modulation of each of these molecules. Typically, the last cellular phenomenon, which is called the metabolism, is heavily regulated by the cellular components that are above it, okay. So what we need to do is not only study these molecular events, that is replication, transcription, translation, and metabolism individually, but we also need to have a nice idea of how are they integrating together to make the cell work, okay. And that exactly what systems biology does for you. Talk about any kind of omics data or omics knowledge that is available, genomics, transcriptomics, metabolomics, the most, well, it's not most recent one, but it has been there since the time of literature curation. It's called bibliomics, where you study the literature and put in all the information that is available in the literature onto your model. So you combine all these omics onto the level of a network, okay. That is what systems biology does for you. Once you have the network, you convert it into a readily computable format so that you can run various simulation. Here, the blues and the greens are the techniques that you can use. The blues are rather the techniques, and the greens are rather the features of the model that you can explore, as well as you can know more about the data. And the red ones are the last that you see are the various application strategies that you can be involving, okay. So here you combine all the data types, or it depends upon you. If you want to work with only a single data type, use a single data, build the network around it, convert it into a model, use the model analysis techniques to know certain features of the model, and finally you apply that to better understanding of the system, okay. The most comprehensive or the most important part of this particular workflow is the reconstruction, okay, reconstruction of a biological network, or in today's discussion, we will rather focus on the reconstruction and modeling of a metabolic model, okay. So what exactly is this? Excuse me. Metabolic model, prior to building, you have to build a reaction network, which basically represents the biochemistry of the organism. So you're basically including the reactions, the metabolites that participates in these reactions, as well as you include the genes that encode the metabolic enzymes that catalyze these reactions. So let's imagine a system where you have the molecule glucose coming from the outside to the inside of a cell by the help of a transporter, okay. This transporter is then encoded or by the genes which are called the glucose transporter proteins. This particular glucose, once it enters the cells, it then gets phosphorylated by an enzyme. It could be a glucokinase in this case, or it could be hexokinase. Okay, so this is your system. So what you basically do is you convert that into the form of a reaction. Okay, and this is essentially the major component of your reaction network. Okay, you capture the metabolites typically with the cellular localization. You capture the genes that encode the enzymes catalyzing these reactions. Okay, so the three most important part of the reaction network or the metabolic network are the metabolites, the reactions, and the genes. Okay, to show you the typical network figure, network basically comprises of nodes and rings. In case of a metabolic network, the nodes will be the metabolites and the links will be the common joining parts that are the reactions. Okay, if you think about a genetic network or a protein-protein interaction network, simultaneously your nodes will be changing from genes to proteins and the links will be their interactions between them. Okay, clear everyone? Okay, so let's move on to the next important topic that we have, that is the most comprehensive available human specific metabolic network, which we call as the Recon 3D. Recon evolves from two different predecessors, okay, that existed before it. We first had Recon 1 that got published in 2007. Recon 1 was significantly extended to Recon 2, which came in 2013. Most recently in 2018, almost 2019, we have the Recon 3D. This is the most updated version of the human metabolic network. Okay, and what exactly is it? Recon 3D is nothing. It's the resource or it's a collection of human specific metabolic reactions. Okay, as you can see, it captures around more than 13,000 human specific reactions and more than 4,000 human specific metabolites. So we'll discuss a bit about how was this built. Okay, as I said, most of the knowledge, apart from the bibliomics or the knowledge that exists in the literature, apart from that, we significantly use the available data. So Recon was built from proteomics, metabolomics data, and all of these data types, but the significant contribution came from the metabolomics data types. We integrated around 10 to 15 different sets of metabolomics data to come to Recon 3D stage. Okay. So what are the challenges that you face usually when you go about building a generic network? So please remember, Recon is generic because it captures the metabolism across the different cell types. There is no cellular or cell specificity maintained in Recon. You will find reactions for liver, you will find reactions for brain, for kidney, all the organs that you have in the human body, their corresponding metabolism or the metabolic pathways, are captured onto Recon. Okay. So what are the challenges one comes across while building a generic network? First, what you do is, let's say we started out with the metabolomics data. We mapped the metabolomics data onto existing Recon. We came to know that there are, let's say, more than 200 metabolites that are missing, that needs to be incorporated onto the current version. So we start with the first metabolite. This is the sphingosine. It's a part of the phospholipid pathway coming from the lipid metabolism. So you include the reaction, okay, corresponding or that involves this particular metabolite from the reaction. Okay, so these are the steps, okay, for you acquire the data, that is metabolomics data, you process it, that is you have to convert the data onto the level of model inputs or reconstruction inputs, okay. You identify both the synthesis and the degradation pathways, and then you add them onto the network. So from a single reaction, you end up having a metabolic pathway involving that particular metabolite. And assembling all these pathways together gives you a wholesome picture of the entire human metabolism. And this is Recon 3D. Okay, you start up with one and you end up having more than 4,000 metabolites incorporated onto this. Okay, so this is the statistics as to what was added. More than 6,000 new reactions were formulated and added onto the current version. Once you have all the additions that you have done to your network, what do? You do run certain quality assurance and quality control tests. Okay, you check for flux consistency. More about it in the next slide, so don't get confused. You also check whether the network is functional, whether the network carries or the reaction carries flux, whether it is active. Okay, that's the time where you also add in the relevant transporters as well as the gene information. Okay, this is very, very important for future simulations. You need to add the corresponding genes that encode the enzymes catalyzing the newly added reactions. Okay. Okay, so once you have this reconstruction, how do you convert it into a model? The first step that you do is you convert it into a stoichiometric matrix. Okay, so here, let's say you have a system where there are two reactions, reaction 1 and reaction 2. You capture the stoichiometry or the coefficient of each metabolite. The metabolites will be taking the row space and the reactions will taking the column space. And by definition, any metabolite that is consumed takes in a negative coefficient and any metabolite that is produced takes in a positive. Okay, if it is not occurring in that particular reaction, it takes in a 0. Okay, that's where you have the stoichiometric matrix. Once you have this matrix, then you define the boundaries, that is the inputs and the outputs for your system. What are my medium composition? Let's say if you're building trying to build a model for a microorganism, you give specific nutrient medium to your microorganism. So that particular nutrient medium becomes your input. Given this particular input, what are the possible outputs or secretion that should come out of this particular organism becomes your outputs. Okay, that's how you define. So inputs are easy to define. Outputs, you just define based upon your experience, okay, or from the available literature knowledge. Okay. After that, you formulate the physiologically relevant objectives and constraints. This is the most important part, where you define what are my conditions under which my model should be active. One of the most important constraints that we follow is the mass balance constraint. Okay, that is S here is the stoichiometric matrix. V is the flux through each of these reactions. The unit of flux is millimoles per gram dry weight per hour. If we have to give a definition for the flux, that is, let's say here A is getting converted. Okay. A is getting converted to B. So the flux will be the amount of B that is produced in this particular reaction. What is the flux through this particular reaction? 1 is the amount, that is millimoles of B produced per gram dry weight of the cell per hour. Okay. That is your flux. So you multiply the matrix across the flux vector, it should be 0. So input equals the output. That is what is maintained thoroughly across these constraint-based metabolic models. Okay, there are various other constraints that you can also put in depending upon the availability of data again. Okay, moving on to constraints, then you define your objectives. Okay, now why do you define an objective? Because you have to say whether your system is functional or not with respect to that particular objective. Most of the time, it is the biomass reaction or the growth rate reaction that takes account whether your system is in a live state or not, whether your cell is growing or not is what is calculated in the biomass objective function. Okay. So what is the use of all these physiologically relevant constraints is once you go on adding these constraints, you end up having a bounded space, and we use the principles of linear optimization to find one of the optimal solution across the edges of this bounded solution space. Okay. Excuse me. So this is the principle also behind the most popular constraint-based analysis method, which is called the flux balance analysis. Okay. moving on, just to give you a brief idea as to what do we mean by biologically relevant. Okay, it is very easy to get the data from the databases as well as the publicly available omics data that is out there. But how do you filter out the biological relevance of all this data that can be used as constraints? Okay, just to give you an example, let's say your objective is production of energy from glucose. This is one of the most commonly used objective, whether it is the microorganism or the human model that you're building for. Okay, so to meet this objective, the constraints or the conditions are, of course, there should be presence of glucose and oxygen, because we know that the aerobic oxidation yields maximum amount of energy compared to that of the anaerobic condition. Okay, but again, the other important constraints that we usually ignore, that glucose can be converted to energy only when you have the transporter protein that will take the glucose from the outside to the inside of the cell, followed by its complete oxidation pathway. When you have the complete oxidation pathway without any gaps, then only can glucose be fully converted into energy. Okay, so this is exactly what I mean by biological relevance. You don't simply put your constraints as per the data, but you attach a biologic meaning to those constraints and objectives. Okay. Recon was tested for, or Recon 3D that I just showed you was tested for such physiologically relevant objectives. Close to 431 objectives were defined and the model was tested for. And what is the most important take home message from here is that you have to read a lot of literature, curate the literature to formulate the relevant objectives and constraints. Okay, that's how the Recon was tested, or whatever model you build in future from Recon has to have a strong curation of literature behind it. Okay. Some of the applications that are currently being used for Recon is mostly in the areas of cell-specific or tissue-specific modeling. This is the area that we'll be also discussing in a few of the slides ahead. Another hot area over here is the host microbe interactions. Okay, keep Recon as your host. You have the gut microbiome, or you have the skin microbiome, or you have the oral microbiome. How is the microbes talking to that to its corresponding host is what we capture and use it to our advantage. Okay, moving on, I'll give you a test case as to how a cell-specific model for the human retina was derived from data to model a cancer type, which is called the retinoblastoma, which is the cancer of the eye. Okay, this is a model specific to the human retina only. Okay, now what exactly is retinoblastoma? As I said, it's a tumor affecting one or both the eyes. In India, typically in the southern part of India, it's pretty high. We get around 1500 cases per year. It has a genetic basis. It is caused by loss of the RB1 gene function. The pictures that I show you here, these are the different stages of retinoblastoma. Okay. Usually the management or the treatment of this particular cancer type is done by chemotherapy or certain drugs, but if they do not give the desired results, it ultimately leads to enucleation of the eye. So any treatment strategy that you talk for retinoblastoma has got its severe side effects. Okay, so can we use the metabolic modeling to know certain biochemical basis for this particular cancer type that can be used to suggest novel therapeutic strategies? That was the target that was set for this particular study. These are certain objectives. As I said, we first have to build a model. We have to characterize the what are certain biochemical features specific to retinoblastoma versus the normal human eye. Is it possible to validate few of our modeling predictions by experimental validation? And then, of course, to predict certain novel drug targets. Okay. Now, as I discuss certain challenges in building a generic network like Recon, I'll also discuss certain challenges in generating any tissue-specific metabolic model. Okay, so usually the starting point for building a cell-specific or a tissue-specific model is Recon, because we treat Recon as a library of human-specific reaction. So from your library, excuse me, you derive your cell-specific model. So what you have is the starting point is the data. It could be any data. It could be, here in this case, the transcriptomic data was used. Okay, and we used one of the model building algorithms to extract the reactions corresponding to this particular data type only from Recon. Okay, so you can replace transcriptomic data with proteomic, metabolomic, any kind of omics data, and you can use different kinds of algorithms. There are at least 10 different kinds of model building algorithms currently available to extract your tissue specific reactions from a generic network like Recon. So using the algorithm, what you end up is a preliminary reaction network. Okay, but please remember, this is a preliminary reaction network, so it have certain inconsistency. Typically, the pathways will not be complete. Transport reactions and most of the exchange or the input outputs will be missed at this stage. So that is the time you read the literature and fill up these gaps. And please remember, any kind of omics data gives you only a snapshot. It only captures only a fraction of the tissue specific functions. So whatever data type you use, you end up finally in reading the literature to fill up your network gaps. And that was what was done. We read a lot of literature to curate our models, to rectify our models, to end up having the finalized version, as well as come up with 50, more than 50 retina specific objectives, again biologically relevant objectives, that were again tested against the model. Okay, all of this curation finally led to the finalized version of the model. As you can see, the total number of reactions will be much smaller because it's only a fraction of the human functionality. So you are extracting a small organ specific model or a tissue specific model from a generic network. Okay, so the number of reactions will be relatively smaller. Okay, so what we then did was, given the different kinds of, so we got the data, if I go back, we got the data is over here that were very specific to the patient type. So we built patient specific retina models. Okay, and all those retina models were tested for the functions that we defined. Those more than 50 retina specific functions were tested when in healthy versus cancer condition. What is happening to these functions? On the y-axis is what you see are the retina specific functions. Okay, here these columns that you see across the x-axis are the patient specific or the retinoblastoma specific metabolic models that were developed and compared across the normal retina model or the healthy retina model. So anything that you see in green is where a retinoblastoma outperforms a healthy retina model. Anything that you see in red is where a healthy retina model outperforms the retinoblastoma models. Anything that in yellow, that means there is no difference. Okay, so to give you a summary of the novel functional insights that we got having this comparison done, what we saw is, compared to the retinoblastoma or the cancer models, the sorry we see a preferential usage of amino acids. These group of amino acids, glutamate, arginine, and ornithine, were preferentially used by the cancer models versus the normal models. So the cancer models were mostly dependent on amino acids versus the normal models were mostly dependent upon carbohydrate for their energy usage. Okay. What we also saw in the cancer cells is there is underutilization of cholesterol. Okay, so if you look into the flux through the cholesterol pathway, the way the cholesterol was synthesized by the normal cells was significantly different from that of the cancer cells. And why was this happening? Because if you come across synthesis of cholesterol pathway, you see a utilization of a redox potential, which is called the NADPH. So what the cancer cell was doing was underutilization of cholesterol, it was preserving the redox potential. So when the cell preserves the redox potential, it kind of masks itself from the antioxidant activity or the defense mechanism of the cell. That's what the cancer cell was doing. Okay. And another very important observation that we saw, which also matched with the literature finding, was that the cancer cells were very specific to their synthesis of essential fatty acids. When we went into literature and identified what does this mean, it was actually a diagnostic strategy that was being used to stratify the patient types across retinoblastoma retinoblastoma cases. Okay, so by synthesizing a specific fatty acid, you mark a specific kind of retinoblastoma type. Okay, and that was also captured onto these models. Okay, once we have an insight of what is happening to the internal biochemistry in the retinoblastoma versus normal, we were next interested, can we use the secretion pattern or the secretion profile for certain validation? Okay, the validation was done by using the mass spectrometry. And then we next perform the drug target analysis testing. Okay, so this is what I show you is the secretome of the retinoblastoma and the metabolic class of the metabolites that were secreted specifically by the cancer cells. Okay, and we were able to validate that yes, whatever the model was saying that a retina model was secreting, it was experimentally validated, and we were giving out true predictions. Okay. Next, what we did is a synthetic lethal study. Synthetic lethal is in cancer world is in order to kill a cancer cell, you need to very accurately kill two genes at a time or a pair of genes or reactions at a time. So killing these reaction pairs gives or disrupts the cancer growth, and those reaction pairs are called as synthetic lethals. And we were able to predict that given we were able to predict at least 13 different kinds of synthetic lethals. Okay, and most of them already existed in the literature. That means most of our predictions were true. Rest were cited as novel drug targets that, given blocking of nucleoside and lipid transporters together, could kill the retinoblastoma cell type or the cancer cell type. Okay, so that was one of the important finding that came out of this study. Okay, so that was retinoblastoma, where we not only gained insight of disease mechanism, but we also predicted certain novel drug targets, typically in the lines of therapeutic measures. Now, moving on from a single, now Recon has been used to derive single cell specific or tissue specific or organ specific models. We as a human, we do not function with single organ alone. We need a nice interaction or integration or crosstalk between our different organs in order to function normally and maintain the cellular homeostasis. So that was what the next study I will be discussing about as to how you can use Recon to give the whole body organ resolved models. Okay, what I give you is the gender specific models. Here the male model has been named after the great mathematician William Harvey. Okay, that's the male specific model. The corresponding female whole body metabolic model is called the Harvey. Okay, so moving on, as I said, Recon was always the starting point. We read a lot of literature pertaining to the different organ models that we are interested in building. We integrated again a lot of different data types. We integrated with proteomics data, metabolomics data, and here for the very first time we integrated a lot of physiological data. Okay, so the model that we ended up having is not only a metabolic model but a phenomenological model. Okay, so a lot of biological phenomenon can be studied using this particular model. So start with Recon 3D. You have these data sets as well as the literature findings as your constraints. Use one of the model building algorithms. to generate the draft models. Again, here, these are the preliminary models. You perform a lot of QC/QA tests run to generate Harvey and Harveta. We ran at least more than 100 iterations to correct the finalized models each time and finally have the finished version of the models. Okay, this is a very lengthy procedure because each time we had the preliminary models, we had to go through it, network contents, whether the pathways that were incorporated across the organ types, do they match its functionality or not? Do they match with the literature findings or not? So all of these tests were formulated and run. Okay. So after having run more than 100 iteration steps, finally we gave the 2 molecular level organ resolved physiologically consistent gender specific genome scale models of the whole body metabolism. Okay. As I said, we have the male specific model, which is called Harvey, comprises of 28 organs, and we have Harveta, which is the female specific whole body metabolic model, comprises of 30 organs. Okay, just to give you a picture, here the models were not just lying in their anatomical position, but rather they were integrated according to their biofluid or surrounding biofluid information. So not only will you get 32 unique organs in this particular whole body model, but also you will get the 13 biofluid compartments which were integrated onto the corresponding organs. Okay, so here you see the biofluids, and here I give you the overall statistics of these models. These are the gender specific organs that were incorporated, and the overall reactions were humongous. So please do not attempt to download these models onto your local machine. You're going to crash it. Okay, so we ran all the simulations on the using the supercomputing services. Okay, so please be aware of downloading Harvey and Harveta. Okay, so it has around more than 80,000 reactions and more than 50,000 metabolites. And here I give you the list of the organs that were incorporated, not only the adult organs, but we also included the blood cell types. Okay, the bold ones that you see here, the intestine, adipocytes, liver, and the red blood cell, these are the organs that were already published. So we also use certain pre-existing models. Rest all of the models were built from scratch using the corresponding literature and data types. Okay. So once we have these models, what is the usage of this? We had to run or test functionalities of these models. One of the best ways to capture the functionalities is whether once you simulate the diseases, are the models able to capture the disease specific biomarkers? Okay, and that is what was simulated across the organ specific whole body organ specific models. Sorry, organ resolved models. Okay, we simulated a group of disorders which are called the inherited metabolic disorders. These are metabolic disorders which you see running across a family, and a lot of metabolite biomarkers are used for their diagnosis. So we use those as our gold standard. As you can see, Harvey and Harveta gave a lot of or a very high accuracy with 85% as true predictions for capturing the biomarkers, and whereas Recon gave only 50%. One of the reasons behind this is because if I go back, sorry, because Harvey and Harveta captures the biofluid compartments, and most of these biomarkers are specific to the blood or CSF or the urine or the sweat. Because these are so excuse me, integrated in such an accurate and anatomically correct manner, that's the reason you can identify a lot of true predictions with these diseases with respect to Harvey and Harveta, whereas Recon has only one extracellular compartment. Okay. Next thing what we did was we studied the inter-organ cycles, because as I said, we as a human body, we have a lot of organs. We range beyond 32 organs. Of course, this model captures only 32. Okay, so these organs rather crosstalk, or they exchange metabolites among themselves to operate in a integrated and ideal manner to maintain the homeostasis. And we checked the existence of these inter-organ metabolic cycles. Sorry. So one of the most important metabolic cycles, as you can see, the presence of the glucose-alanine cycle, where liver generates the blood glucose or maintains the level of glucose in the blood, which is taken up by the muscle in case of energy usage, converts it into pyruvate and finally alanine, releases into blood and back into the liver. So presence of these inter-organ or exchange of metabolites between the organs were tested and verified across literature, and yes, these models were able to capture them. Next, what we did was integrated the microbiome, here specifically the gut microbiome information. Okay, so again, we use the data, metagenomics data, for healthy individuals and incorporated information for more than 700 gut microbe specific genome scale metabolic models. Okay, here you can see the most highly represented phyla that being used for microbiome integration. Once integrated, the model size was humongous. Okay, but we were able to capture not only certain pre-existing knowledge with respect to secretion of neurotransmitters by gut microbes that can be used up by the human host, but also gave certain novel predictions with respect to the neurotransmitter, that is GABA and histamine. Okay, so what is the potential here for the whole body organism models is you can use this to study the brain gut axis with respect to integration of the gut microbiome information. Okay, so coming to the summary of my talk was, you have using constraint-based modeling approaches, and you can use Recon, you can use Harvey and Harveta also as your library of information. You can use that for proposing novel therapeutics and also gaining what I usually love about this. It gives us a lot of novel mechanistic insights about the disease condition or any kind of condition that we want to simulate. Okay, and Harvey and Harveta in this case were foundations for virtual human. We are slowly approaching to that level. And very, very important that we used for the retinoblastoma study was, you need to combine the computational studies with that of the experimental validation so that it makes the study much more believable, as well as it can aid in future advancements in your scientific knowledge. Okay, so I want to end my talk citing the kind of approach that one needs to adopt in order to study medicine and for studying disease processes, which we call as the P4 medicine approach. The 4 Ps are the predictive, preventive, personalized, and participatory. Okay, and this is exactly what the strategy for systems biology, typically for the biomedical purposes, is being followed. Okay. Here I show you certain research themes of my lab. Okay, what I showed is how we can use the current network and use certain data types for its expansion, and also use the data, the same data, for its analysis. Most of my other work are involved in studying host and gut microbiome relationship, typically in disease condition, in cancer, as well as in neurological disorders. Okay, I'm also highly involved in studying the effect of diet and medications with respect to human health and disease conditions. Okay, so if any of you are interested, you can write to me. There are open positions also. Okay, and with that, I need to acknowledge my students who did all of the work. Okay, both current as well as former, as well as my experimental collaborators, computational collaborators, that is both within IIT and abroad. Okay, and most of my funding comes from DST Inspire. and of course, a lot of healthy discussions that were had with the group of IBSC. So thank you.